Welcome to another deep dive. I have to say, you sent over some intense notes this time about eukaryotic RNA transcription, processing, and regulation. You weren't kidding when you said you wanted to go deep.
Oh yeah, there's a lot to unpack here.
Yeah.
But it's also fascinating. So many intricate processes happening.
Well, I'm ready to dive in.
Great.
Should we start with the control mechanisms of eukaryotic RNA transcription?
Absolutely. Let's start there.
Perfect.
Uh,
My notes mention these two key players, basal expression and then regulated expression.
Right. So basal expression is all about just making sure that the RNA polymerase II gets to the right spot
Okay.
and that will determine how often transcription starts. And so it includes things like the tita box. That's a big one.
Right.
The promoter, of course. And then things that are near the promoter proximal elements. So all that is just getting things set up and ready to go.
Okay, so that's like the baseline.
Exactly. Yeah.
And then regulated expression kind of takes it to the next level.
Yeah. It responds to different signals.
Okay.
So it could be hormones, it could be heat shock, it could be heavy metals, chemical stimuli, that kind of thing.
Wow.
And these things are going to determine the rate of transcription.
Okay.
It can either enhance it or repress it.
Got it. So it's like the cell is responding to its environment and adjusting accordingly.
Precisely. Yeah.
Very cool. The notes also mention something about this regulated expression acting in both directions and even stemming from introns.
Yes. It's not limited to just upstream sequences.
Okay.
So, we always think of things linearly, but this is kind of showing that, the influence on a gene can be upstream or downstream.
Okay.
And it can include genes that are further away on a chromosome.
Wow.
Yeah.
That's pretty wild.
Yeah.
So it's not just this like linear process, but more like a complex network of interactions.
Precisely. And at the heart of this network, you have your cis elements and your transacting factors.
Okay. Can we define those?
Yeah. So your cis elements, those are DNA sequences that are going to serve as binding sites for your transacting factors. So those are your activators or repressors.
Got it.
And these bind with high specificity, much like a lock and key.
Oh, okay. So the cis element is like the address on the DNA, and then the transacting factor is the mail carrier, either delivering an activation signal or a repression signal.
Exactly. Yeah, that's a great analogy.
And then to make it even more interesting, we also have these coactivators and adapters.
Yeah, so coactivators kind of bridge the gap between your activators and the basal transcription machinery.
Okay.
So they kind of help get everything together. And then you have your adapters, and those can help to bring distant DNA regions close together.
So it's like a team of architects and engineers making sure that the right genes are expressed at the right time.
Exactly, yeah. And then if we compare prokaryotic and eukaryotic transcription factor mechanisms, there's a stark difference there.
Okay.
Prokaryotic systems are much more simple, with transcription factors often interacting directly with RNA polymerase. But in eukaryotes, it's much more elaborate, with multiple layers of regulation and a complex interplay of factors.
Got it. It reminds me of that time I was trying to knock down p53 gene expression using CRNA. It worked beautifully in lung cancer cells, but my student was having no luck in colon cancer cells.
Yeah, and that's because the colon cancer cells, they were tetraploid. And so the p53 gene, it's tightly packed, making it kind of inaccessible to CRNA.
Interesting. So the structure of the chromosome itself can impact RNA transcription.
Absolutely, yeah. It really highlights how cellular context matters.
For sure. Now, I think we need to transition into talking about the assembly of the eukaryotic transcription complex, right?
Right. And so eukaryotic RNA polymerases, they require general transcription factors. We call those GTFs.
Okay.
And that's to recognize promoters unlike their prokaryotic counterparts.
So it's like the RNA polymerase needs a guide to find the right spot on the DNA.
Exactly, yeah. And so the assembly process, it's a multi-step affair.
Okay.
And it starts with this TATA binding protein. We call that TDP, which binds to the TATA box.
Got it. So that's the first one to arrive on the scene.
Yes. And then TFII and TFIA join the complex. Okay. TFIIB, that one's particularly interesting. It recruits RNA polymerase II to the promoter.
Okay.
And it also interacts with TFII, which helps to stabilize the complex.
Oh, so TFIB is kind of like a bridge connecting the RNA polymerase to the promoter.
Exactly.
All right, so we have our TBK, TFIA, TFIIB, and TAFI. What's next?
All right. So then after that, we've got TFIIE and TFIH.
Okay.
TFIH, that one's super important because it has two key enzymatic activities.
Okay. What are they?
First, it acts as a helicase. So it's unwinding the DNA double helix at the transcription start site.
So it's like a zipper opening up the DNA strand.
Exactly.
Okay. And the second activity?
The second one is a kinase activity. So it phosphorylates the C-terminal domain, or CTD, of RNA polymerase II.
And what does that phosphorylation do?
That's going to be crucial for the transition from initiation to elongation.
Okay.
It's kind of like a trigger. It says, okay, we're ready to start moving along the DNA template.
Got it. And I see my notes here talking about this RNA polymerase 6 CTD. It says that it's a unique structure with a repeating sequence of 7 amino acids.
Yes, the heptapeptide repeat is tyrosine, serine, proline, threonine, serine, proline, serine. And the phosphorylation of serine 5 within this repeat, it's really important for the polymerase to get activated and recruit factors that are going to be involved in RNA processing.
Okay, so it's not just turning the polymerase on, but it's also kind of setting the stage for what happens next.
Precisely. It's going to act as a platform, a landing pad for all these different factors that are involved in pre-mRNA capping, splicing, and 3' end processing.
Wow. So it's like a central hub coordinating the whole RNA processing machinery.
Exactly.
Very cool. Okay, I think we should move on to 3' polyadenylation, which is another key process in eukaryotic mRNA processing.
Okay.
And this is where we see a really big difference between eukaryotes and prokaryotes, right?
Right. So prokaryotic mRNA, it doesn't go through polyadenylation.
Okay.
And that's because their transcription and translation, it's coupled. It kind of happens at the same time.
Right.
In eukaryotes, you have transcription occurring in the nucleus and then translation occurring in the cytoplasm, which allows for this more complex processing of the RNA transcript.
Okay, so in eukaryotes, the mRNA gets modified before it can head out to the ribosomes.
Exactly.
Okay, so what happens during polyadenylation?
So eukaryotic mRNAs, they're going to have this polyadenylation signal. It's a sequence, AAUAAA.
Okay.
And it's upstream of the cleavage site.
Okay.
And that signal is recognized by a complex of proteins, which includes cleavage and polyadenylation specificity factor, which we call CPSF, and cleavage stimulation factor, or CSTF.
So these factors bind to the signal and mark the spot where the mRNA needs to be cut.
Precisely.
Yeah.
And CPSF, it recruits polymerase.
Okay.
And that's going to add a tail of adenine nucleotides to the 3' end of that cleaved mRNA. And it can be up to 200 nucleotides long.
Wow. Okay, so what's the purpose of this poly A tail?
It has multiple functions, actually.
Right.
One is to protect the mRNA from degradation by exonucleases.
Okay.
It also enhances its export from the nucleus, and it plays a role in translation initiation.
Got it. So it's like adding a protective cap and a shipping label to the mRNA molecule.
Exactly. Yeah, that's a great way to put it.
Okay, so that's the rule, but of course there's always an exception to the rule in biology.
Right.
What's the exception here?
So histone mRNA. Histone mRNA, it's not going to have a poly A tail. Instead, it has a stem loop structure at its 3' end, and that's going to be formed with the help of U7 snRNP and stem loop binding protein.
Okay, so it's a different strategy, but ultimately trying to achieve the same goal, which is stability and proper processing.
Exactly.
And I also noticed that polyadenylation is related to transcription termination.
It is, yes. So after the mRNA is cleaved, a 5 to 3 month exonuclease called XRN2 comes in, and it degrades the remaining RNA that's still associated with RNA polymerase II. And that degradation is thought to trigger the release of the polymerase from the DNA.
Got it. So it's like one event triggers the next, like a domino effect.
Exactly.
Yeah.
So are we ready to move on to mRNA splicing?
Yes, let's do it. Splicing, this is where we remove the introns, right?
Precisely. Yeah.
Okay. And this is carried out by the spliceosome.
It is. It's composed of RNA and proteins.
Okay.
And the RNA components, those are small nuclear RNAs, or snRNAs, which are associated with proteins.
Okay.
And then those together form small nuclear ribonuclear proteins, or snRNPs.
All right. So it's a team effort with both RNA and proteins working together to get rid of these non-coding regions.
It is, yeah. And so we have 5 major snRNPs that are involved in splicing.
Okay.
U1, U2, U4, U5, and U6.
Okay. So let's break down what each one does.
Yeah.
Let's start with U1. What's its role?
Okay. So U1, it's going to recognize and bind to the 5' splice site.
Okay.
And that site is marked by a GU dinucleotide sequence.
Okay. So it's like the scout finding the beginning of the intron.
Yeah, exactly.
Okay, and then what about U2?
All right, so U2 snRNP, that's going to bind to the branch point,
Okay.
and that's an adenine nucleotide that's located within the intron.
Okay, so it's marking a specific spot within the intron.
Yes.
What happens next?
Okay, so then a complex of U4, U5, and U6 snRNPs, it joins the party, forming the inactive spliceosome.
Okay,
And then that complex undergoes a bunch of rearrangements, leading to the release of U1 and U4, and then you have the formation of the active spliceosome.
got it. So it's like assembling a puzzle. Each piece has to fit in the right order.
Exactly. And then in the active spliceosome, the branch point adenosine attacks the 5' splice site,
Okay.
which breaks the phosphodiester bond and then forms a lariat structure from the intron.
So the intron gets looped out, forming this lasso-like structure.
Exactly. And then the 3' OH group of the upstream exon, it's going to attack the 3' splice site.
Okay.
And that joins the two exons and releases that intron lariat.
Wow. It's a beautifully orchestrated process.
It is, yeah. And the splicing reaction, what's interesting is that it's catalyzed by RNA molecules themselves.
Oh, wow.
So we call those catalytic RNAs ribozymes. And it really highlights just how versatile RNA is.
So RNA can be both the blueprint and the builder in this process.
It can, yes.
Incredible. Now, I know that this nuclear mRNA splicing is the most common, but there are other types of splicing as well.
Right. So you have group I and group 2 introns, and those can self-splice, which means they don't need that spliceosome machinery.
Wow, so they can just cut themselves out.
They can.
That's amazing.
Yeah. So group I introns, they're going to use a guanosine cofactor.
Okay.
And then group 2 introns use an internal adenosine to initiate the splicing reaction.
Okay. So they have different strategies, but they're both trying to achieve that same goal of getting rid of the intron and joining the exons.
Exactly.
Okay. And then we also have tRNA splicing,
We do, yes.
which involves a different set of enzymes.
It does.
Okay. So it seems like every type of RNA molecule has its own unique processing pathway.
It does, yeah.
That's incredible. Okay, I think we should move on to tRNA maturation now, right?
Okay, sounds good.
This is another really important process in gene expression.
It is.
And this is the molecule, tRNA, that carries amino acids to the ribosomes during protein synthesis.
It is, yes. And tRNA molecules undergo a series of processing steps to become functional.
Okay, what are those steps?
Well, the main ones are 5' and 3' end cleavage, and then splicing if there are introns present.
Okay.
And base modification, of course. And then one of the key steps is the addition of the CCA sequence to the 3' end of the tRNA.
Okay, and this CCA sequence, this is where the amino acid is going to get attached.
Precisely.
Okay, and what catalyzes that addition?
That's going to be tRNA nucleotidal transferase.
Wow. It's amazing how every step in this process is so carefully orchestrated.
It is, yeah.
Now, I want to go back to this group I and group 2 cell splicing for a second,
Okay.
because you said that the intron itself acts as the catalyst. Can we explain that a little bit more?
Yeah, sure. So in group I cell splicing, it uses an external guanosine as a cofactor.
Okay, so this guanosine molecule comes in from outside the intron.
Right, and the 3' OH group of that guanosine is going to attack the 5' splice site. And that initiates a bunch of transestrification reactions that lead to the removal of the intron and the joining of the exons.
Wow. So it's like a molecular dance with these bonds breaking and forming in a very specific order?
It is. It's a very elegant process.
That's incredible. Now, how is group 2 self-splicing different?
So group 2 self-splicing, it's a little bit different because it doesn't need that external cofactor.
Oh, okay.
Instead, it uses an internal adenosine that's within the intron.
Oh, so the intron itself has everything it needs.
It does, yes. So the 2' OH group of that adenosine, it attacks the 5' splice site, and that initiates the splicing reaction. And group 2 self-splicing also forms a lariat structure, similar to what we saw with the nuclear mRNA splicing.
Okay, so a similar outcome, but a different mechanism.
Exactly.
Very cool. All right, I think we need to talk about alternative splicing now.
Okay, sure.
This is a key mechanism for generating protein diversity, right?
It is, yes.
From a single gene.
From a single gene, yes. And so alternative splicing, it allows for a single gene to produce multiple protein isoforms,
Okay,
which are going to have distinct functions.
got it. So it's like having a master blueprint that can be customized to create different versions of a protein.
Exactly.
Okay. And there are 4 main types of alternative splicing, right?
Right. You have exon skipping,
Okay.
alternative 5' splice site selection,
Okay.
alternative 3' site selection,
Okay.
and then intron retention.
Okay. Let's break those down a little bit,
Yeah.
starting with exon skipping. What's that all about?
So exon skipping is the most common one, and it involves the exclusion of an exon from that final mRNA.
Okay. So it's like removing a chapter from a book. You get a shorter version of the
Yep.
story.
Exactly.
Okay. And then alternative 5-on-3 splice site selection.
Yeah. So that's going to involve choosing different splice sites within an intron,
Okay.
and that can lead to the inclusion or exclusion of parts of exons.
Oh, okay. So it's like having different options for where to cut and paste the genetic code.
Exactly. And then intron retention, that's the least common one.
Okay.
And that's where an intron is actually included in that final mRNA.
Oh, interesting. So it's like keeping a deleted scene in a movie.
It is, yeah.
And all of this is regulated, of course.
Yes, by a complex interplay of cis-acting elements and transacting factors.
Okay. So it's back to our addresses in mail carriers, but
Yeah.
this time they're controlling which exons get included in the final message.
Exactly. And it allows for such a remarkable level of flexibility in gene expression.
It's incredible how much complexity can arise from a single gene.
It is, yeah.
So I guess we should touch on the emerging applications of mRNA in biotechnology. Things like CRISPR-Cas9 technology and RNA vaccine.
Right. So CRISPR-Cas9, it utilizes a guide RNA to target specific DNA sequences,
Okay.
which allows for a very precise gene editing. And then RNA vaccines, those deliver mRNA that encodes for viral proteins into cells, which triggers that immune response and protects against infection.
Got it. So in both cases, we're using RNA to manipulate cellular processes for therapeutic purposes.
Precisely. And researchers are really actively exploring, how safe and effective are these RNA-based therapies?
Yeah. It's an exciting field with a lot of potential.
It is, yeah.
All right. Before we wrap up this first part of our deep dive, maybe we should transition to thinking about mRNA's critical role in protein synthesis and gene expression. This seemingly simple molecule is such a vital go-between, carrying that genetic information from DNA to the ribosomes.
It is, yeah. It's like a messenger,
Yeah,
delivering those instructions from the nucleus to the cytoplasm.
exactly. And it's the sequence of codons within the mRNA that's going to determine the order of amino acids in the protein.
Right. And each codon, that triplet of nucleotides, is going to correspond to a specific amino acid. And that genetic code is universal.
So it's a language that all cells understand.
Exactly. And then translation, that's where the mRNA is decoded into a protein. It involves ribosomes, tRNA, a bunch of protein factors. It's this elegant molecular dance.
Right. And tRNA acts as that translator, bringing in the correct amino acids based on the codons in the mRNA.
Exactly. And ribosomes bind to the mRNA, and they move along, reading those codons one by one.
Okay.
And then tRNA molecules carrying specific amino acids, they're going to recognize and bind to their corresponding codons on the mRNA, and that ensures that the amino acids are added in the correct order.
That's incredible. It's this amazing process with all these different molecules working together to translate the genetic information into a functional protein.
It is. It's a fundamental process of life.
Wow, we've covered so much ground already. It's really clear that RNA plays a central role in gene expression.
It does, yeah.
And this is just the beginning.
It is.
There's so much more to explore in the RNA world. I'm really looking forward to diving into the different types of RNA molecules and their specialized roles next time.
Yeah, it'll be fun. I'm looking forward to it. Welcome back to our deep dive into RNA.
Yeah, we're ready to keep going.
You gave us so much material. It's incredible how much there is to talk about.
It is, and we're just getting started.
I know. It's amazing.
So last time we focused mostly on mRNA,
Right.
but I know that there are so many other kinds of RNA molecules in the cell.
There are. Yeah,
What other ones should we talk about?
so many.
Yeah.
The world of RNA is very diverse, and each type of RNA molecule has a very particular and specific role in the cell.
Okay.
Why don't we start with ribosomal RNA,
Okay.
rRNA?
rRNA.
And that's a major component of ribosomes.
Right, the protein synthesis machines.
Exactly.
It's interesting that RNA is such a key part of what builds proteins.
It is, yeah. It makes up a significant portion of the ribosome's structure,
Okay.
and it provides that framework for protein synthesis.
Okay.
But what's even more fascinating is that RNA itself has catalytic activity.
Oh, wow.
So it plays a direct role in peptide bond formation during translation.
So it's not just a scaffold, it's an active participant.
It is.
That's amazing.
And then we have, of course, transfer RNA,
tRNA.
tRNA,
Yeah.
and that's essential for translating that genetic code in the mRNA into the language of amino acids.
They're like the adapters.
Right.
They link the codons in the mRNA to the corresponding amino acids.
Precisely. And so each tRNA molecule has an anticodon,
Okay.
which is that triplet of nucleotides that's going to base pair with a particular codon on the mRNA.
Okay.
And then on the other end of the tRNA, you have the attachment site for the amino acid
Right.
that corresponds to that codon.
Makes sense.
So that ensures that the right amino acid is added during translation.
Yeah, it's like a delivery system, making sure that the right parts get to the right place
Precisely.
at the right time.
Exactly.
Okay, so those are important for protein synthesis.
Right.
But I know there are other RNA molecules that do other things in the cell,
There are.
not just protein synthesis.
Right. Let's move beyond protein synthesis and think about some other fascinating roles that RNA molecules play.
Okay.
So we already talked about small nuclear RNA, snRNA.
Right, for splicing.
Yeah, in the context of mRNA splicing.
Right, they're part of the spliceosome.
Exactly.
Okay.
And they work together to recognize those splice sites, bring them together, and then catalyze the splicing reaction.
Right.
And some snRNAs can even act as ribozymes, as we talked about before.
Right, so they can do the catalysis themselves.
They can, yeah.
Amazing.
Another interesting class of RNA molecules are the small nucleolar RNAs, or SNRNAs.
Snornase.
And these are primarily located in the nucleolus.
Which is in the nucleus.
Yes, a dense region within the nucleus.
Okay.
And they guide the chemical modifications of other RNA molecules,
Okay.
particularly RNA and tRNA.
Okay.
So they act as guides,
Okay.
directing enzymes to specific sites on those RNA molecules.
Right.
And they can direct methylation or pseudoderylation.
So they're like the quality control team,
They are, yeah.
making sure that everything is modified correctly so that
Yeah.
they function properly.
Exactly.
Okay, what else?
All right, let's talk about microRNA, or miRNA.
Muvernan.
These are tiny, typically only about 22 nucleotides long.
Wow.
But they've emerged as key regulators of gene expression.
Okay, I've heard about these. They can silence genes.
Yes, they function by base pairing with target mRNAs, usually in the 3' untranslated region.
Okay.
And that binding can lead to either mRNA degradation or translational repression,
Okay.
which is going to reduce the production of the protein.
So it's like a dimmer switch
Exactly.
that can fine-tune the expression of certain genes.
It is.
Yeah.
And these have been implicated in a wide range of cellular processes: development, differentiation, cell proliferation, apoptosis,
Wow.
and dysregulation of miRNA expression has also been linked to several diseases, including cancer.
So these tiny little molecules
Huge impact.
have such a big impact.
Yeah.
It's amazing.
So we've talked about different types of RNA molecules,
Right.
but we also need to think about the mechanisms that regulate RNA processing and stability,
Okay.
because those are really crucial for making sure that the right RNA molecules are made at the right time in the right amounts.
Right, like a quality control system. To
Exactly.
make sure everything is working correctly.
Yeah.
Okay, so what are some of those mechanisms?
Well, one important regulatory mechanism is RNA editing.
RNA editing.
And that involves modification of RNA sequences after transcription.
Okay.
So it's going to alter the information that's encoded by the RNA molecule.
So it's like editing a manuscript after it's already been written.
Exactly.
Okay, so what kinds of modifications happen?
So you can change the coding sequence of an mRNA, and that leads to the production of a different protein isoform.
Okay.
You can affect splicing patterns.
Okay.
You can alter the stability of the RNA molecule.
Wow, so many different things.
Yeah, so much flexibility in RNA processing.
Amazing.
Another key regulatory mechanism is RNA interference,
Okay.
or RNAi.
RNA, I've heard of that.
And this is triggered by double-stranded RNA molecules.
Okay.
And it can lead to degradation of target mRNAs or inhibition of their translation.
So it's like a targeted attack
It is, yeah.
against specific RNA molecules.
It's like a cellular immune system that targets specific RNAs.
Exactly. That's a great way to put it.
And it's become such a powerful tool for regulating gene expression.
Yeah.
And also an important research tool to study gene function and even develop new therapies.
So it's both a natural process and a technology.
It is, yeah.
That's incredible.
Now, we also have to talk about non-coding RNA,
Okay.
or ncRNA.
NC.
And these, unlike mRNA, don't code for proteins,
Right.
but they still play really important roles in the cell.
Okay.
And they're involved in a wide range of processes, including gene regulation,
Okay.
chromatin remodeling,
Wow.
even protein trafficking.
Really?
Yeah.
Okay.
One example that we can talk about is long non-coding RNA.
LNCRNA.
LNCRNA, yes. And these are typically longer than 200 nucleotides.
Okay.
And they can regulate gene expression through various mechanisms.
Okay.
They can act as scaffolds, bringing together proteins that regulate transcription.
Oh, okay.
They can act as decoys, binding to and sequestering transcription factors, or even miRNAs.
Wow, so they're like master regulators,
Yeah.
affecting the expression of multiple genes.
Yeah.
Interesting.
We also have circular RNA
Circular R
for siRNA.
for RNA.
And these, as the name suggests, form a closed loop
A
structure.
closed loop. How does that even happen?
Right.
I thought RNA molecules had like a 5-foot end and a 3-foot end.
They do, but these are formed through a backsplicing event.
Okay.
So a downstream 5-foot splice site is joined to an upstream 3-foot splice site.
So the RNA molecule basically loops around and splices itself into a circle.
It does.
That's so cool.
And this makes them very stable because
Never-
they're resistant to degradation.
So they hang out in the cell for a long time.
They do.
Okay, what do they do?
Well, they've been shown to act as sponges for myrinase.
Okay.
So they bind to them
Okay.
and sequester them, which reduces their activity.
So they regulate the regulators.
Precisely.
Wow.
And the world of ncRNA is really still largely unexplored, and scientists are constantly finding new types
Wow.
with novel functions.
That's a whole world in itself.
It is. It's a rapidly evolving field.
That's so cool.
I think we should move on to RNA therapeutics.
Okay.
What do you think?
Yeah, it seems like RNA is a hot target these days for drug development.
It is.
Okay.
RNA-based therapies have such great potential for treating a wide range of diseases.
For sure.
One promising approach is RNA interference, RNAi, that we talked about earlier.
Right, silencing those disease-causing genes.
Exactly.
Okay.
By introducing synthetic double-stranded RNA molecules that target specific mRNAs.
Okay.
It can reduce the production of the proteins.
Right.
And this is being explored for cancer, viral infections, lots of different things.
Wow.
What other RNA-based therapeutic approaches do we have?
Yeah. What else?
Well, another really exciting area is mRNA-based therapy,
Okay.
where synthetic mRNA molecules are delivered to cells to produce a therapeutic protein.
So we're giving the cell the instructions to build what it needs.
Precisely.
Okay.
And this has shown great promise in treating genetic disorders where a functional copy of the gene is missing.
Right.
By delivering that correct mRNA,
Okay.
you can restore the production of that missing protein.
That's amazing.
And then, of course, RNA vaccines.
All right, the mRNA vaccines.
Which have been so effective against COVID-19.
Right.
So RNA vaccines are delivering mRNA that's encoding those viral proteins into cells.
Right.
And then the cells produce the viral proteins.
Okay.
And that triggers that immune response that protects against future infection.
Yeah, it's an amazing approach.
It's really revolutionized vaccinology.
It has.
Now, this is a little bit of a tangent, but I think it's really interesting to think about RNA's role in evolution, because many scientists think that RNA played a central role in the early stages of life on Earth.
The RNA world hypothesis.
Exactly.
I've heard of that.
Yeah, the idea that RNA, not DNA, was that primary carrier of genetic information in early life forms.
Right.
And that's because RNA has this amazing ability to both store genetic information,
Okay.
but also catalyze biochemical reactions.
Right.
So that suggests that RNA could have been both the blueprint and the builder
That's amazing.
in early life.
So then DNA took over information storage,
Right.
and proteins took over most of the catalysis.
Right.
But RNA is still essential
Is.
for so many things
For so many things.
in modern cells.
Yeah, it's that mediator
Yeah.
of the flow of genetic information
Oh.
from DNA to protein. It
Yeah.
regulates gene expression.
Right.
It catalyzes key reactions.
It does it all.
It's a testament to how versatile it is.
It is.
And before we wrap up this part, we
Yeah.
should touch on RNA viruses. These are viruses that use RNA as their genetic material, unlike most organisms.
Okay.
And they've evolved these remarkable strategies to replicate and spread.
Okay.
And they often cause disease.
Right.
So things like influenza virus,
Yep. Right.
HIV,
Right.
coronaviruses.
Yeah, these are formidable opponents for sure.
They are.
We need to understand them to come up with treatments.
Yes. Understanding how they work is key for developing those antiviral therapies and vaccines.
Absolutely.
So that concludes part 2 of our deep dive into the world of RNA.
Wow, we covered a lot of ground.
We did.
From different types of RNA molecules to their roles in cellular processes
Right.
and their potential for therapeutic applications.
Yeah.
It's incredible.
And there's still so much more to explore.
Yeah, I can't wait for part 3.
Yeah, me too.
Welcome back to our deep dive into the RNA world. It's amazing how much we've already covered, from transcription and splicing to the diverse roles of all these different RNA molecules.
Yeah, it's pretty incredible, and we've only just scratched the surface, really.
Mhm.
The field of RNA biology is just constantly expanding with new discoveries coming out all the time. It seems like we're always learning more about the complexity and versatility of this amazing molecule.
So what's next? What are the frontiers of RNA research right now? What are scientists most excited about these days?
Well, one area that's generating a lot of excitement right now is the study of RNA modification.
RNA modifications. Okay.
Yeah, for a long time, we've been so focused on the four canonical bases, adenine, guanine, cytosine, and uracil. But now we're realizing that RNA molecules can actually be decorated with all sorts of chemical modifications. It's like a whole new level of complexity.
So it's not just about the sequence of the bases, but it's also about these chemical tags that can be added on top of that.
Exactly. And these modifications, they can influence a lot of things. RNA stability, localization, translation, even interactions with other molecules.
Wow.
It's like they're adding another layer of meaning to the RNA language.
That's so cool. Can you give me some examples of these modifications?
Sure, one of the most well-studied ones is N6-methyladenosine, or M6A.
I'm 6A, okay.
And that one's found in a wide range of RNAs, and it plays a lot of different roles in regulating gene expression.
Okay, so how does it affect gene expression?
Well, it can influence mRNA stability, splicing, even translation efficiency.
Wow.
For example, it's been shown to play a role in regulating the circadian clock
Oh, wow.
by influencing the stability of certain mRNAs that are involved in that process.
So it's like fine-tuning those protein levels to keep the cellular clock running smoothly.
Exactly. Another interesting modification is pseudouridine.
Okay.
That one's denoted by the Greek letter E, and that one's been found to enhance the stability of certain RNAs,
Okay.
like tRNA and rRNA.
So it's like adding extra reinforcement to those key players.
Exactly. And, the field of RNA modifications is still pretty young, but it's already clear that these chemical tags play a really critical role in regulating RNA function.
It's amazing to think that we're still learning so much about this fundamental molecule.
It is.
What are the implications of understanding these RNA modifications? What can we do with this knowledge?
I mean, the implications are huge. I think understanding how these modifications work could lead to all sorts of new ways to manipulate RNA function for therapeutic purposes.
Okay.
Imagine being able to selectively modify RNA molecules to treat diseases.
It's like having a molecular toolkit for RNA engineering.
Exactly. Another really exciting frontier in RNA research is the development of these single molecule techniques for studying RNA.
Single molecule technique.
Yeah, okay.
These techniques allow scientists to actually visualize and manipulate individual RNA molecules
Wow.
in real time.
So we can actually watch RNA molecules in action.
Yeah.
That's incredible.
And these approaches, they're giving us so much insight into the dynamics of RNA folding,
Okay.
interactions with other molecules, even the mechanisms of RNA-based enzymes. It's revolutionizing our understanding of how RNA works at the most fundamental level.
It is. It's really amazing.
It is. Okay, so we've talked about these exciting new areas of research,
Right.
but I want to shift gears a little bit and talk about RNA-based technologies. What are some of the applications that are emerging from this field?
Right. Well, one area that has a ton of promise is RNA therapeutics, which we touched on a bit earlier.
Right.
RNA interference, or RNAi, it's already being used to develop treatments for a bunch of different diseases.
Okay, so that's that silencing of the disease-causing genes with this amazing precision.
Exactly. And mRNA-based therapies, those are really gaining traction too. I mean, the success of the mRNA vaccines against COVID-19, it's really demonstrated the potential of this approach.
Yeah, for sure. It's like we're turning our cells into these little protein factories that produce these therapeutic molecules on demand.
Exactly. And beyond treating diseases, we're also exploring RNA-based technologies for diagnostic purposes. So RNA biomarkers, those can provide really valuable insights into disease states, allowing for earlier and more accurate diagnoses.
So it's like having a molecular fingerprint that can tell us about the health of our cells.
Exactly. And in biotechnology, RNA-based tools are being used for all kinds of things: gene editing, crop improvement, even the development of new materials.
It seems like RNA is becoming this versatile tool for engineering biology.
It is. The possibilities really seem endless.
They do. This deep dive has been incredible. I'm just blown away by the complexity and versatility of RNA.
Me too.
It's amazing.
And as we continue to explore the RNA world, I'm sure we'll uncover even more wonders and possibilities.
I think so too. It's such a fascinating field.
It is. Well, I'm already looking forward to our next deep dive. But until then, keep exploring, keep questioning, and keep marveling at the intricate beauty of the molecular world. Thanks for joining us.